Sarcoidosis is a rare disease caused by inflammation. It usually occurs in the lungs and lymph nodes, but can occur in almost any organ, and these are the different organs that can be affected in their respective percentages. Pulmonary sarcoidosis, which is the topic of interest, is prevalent in 90% of the case. The disease generally affects of any age with peak incidence in individuals from 20 to 40 years and women showcasing a second peak incidence at 65 to 69 years. The disease is characterized by 9 necrotizing granulomas, which are sites of inflammation and an accumulation of CD4+ T cells and macrophages. Interestingly enough, there are individuals who are genetically susceptible to sarcoidosis, as seen with their HLA genes genotype. HLA molecules are expressed by antigen-presenting cells and are responsible for presenting antigens to T cells. The DRB subtypes, particularly the HLA DRB1-0301 type, suggest increased risk to sarcoidosis. The unknown antigen is presented by the HLA molecules to the gene-expressing T cell receptors which are accumulated in this phenotype. It has been found that the TCR-HLA complex forms a peptide binding pocket that perfectly matches a vimentin-derived peptide, a suspected antigen. High affinity binding interaction of the TCR and the antigen induces clonal expansion that mounts a hypersensitive immune response. The disease is still a problem worldwide because there are no reliable biomarkers found yet to diagnose the disease, which often leads to a lot of false positives. The disease mostly affects African Americans, North Europeans, and individuals with predisposition. Currently, no specific diagnostic tests exist for the disease. The suggested biomarkers are not specific, as the severity and progression of the disease remains unnoticed. If the identification of a reliable biomarker is not improved, the disease will continue to increase, leading to mortality. We propose a diagnostic test that combines immunohistochemistry and quantum dots, which considers the secretory phenotype of macrophages involved in granuloma formation, and the TCR-HLA complex in the HLA gene found in individuals that forms a peptide binding pocket that perfectly matches a suspected autoantigen, which is the vimentin-derived peptide. Finding a better way to diagnose the disease and a more reliable biomarker will improve good health and well-being in our communities, which will subsequently result in sustainable cities and communities. The techniques that are going to be used in the project are sandwich ELISA, immunohistochemistry, indirect ELISA, and fluorescent microscopy. Perform bronchoscopy on individuals to obtain BALF. Coat the well with gene-expressing TCRs and incubate. Add BALF, wash to remove unbound contaminants. Add HLA gene molecules conjugated with quantum dots. Visualize using fluorescence microscopy and perform 3D modeling to identify the antigen. For indirect ELISA, tissue samples were collected and processed. To prevent non-specific binding and improve antibody penetration, a blocking buffer was used. Primary antibodies specific to CD receptors of macrophages were added to the tissue sample. Secondary antibodies quantum dot conjugated were added to bind to the primary antibodies. Using a fluorescence microscope, the attachment to the target receptors was then visualized. The total amount that will be required for the research proposal amounts to 210,000 rands, and the project will proceed for 3 to 5 years. Increased QD fluorescence in the experiment compared to the control would suggest binding of vimentin to the T cell receptor complex, suggesting a new biomarker. Would also suggest a high ratio of M2 macrophages to M1, for differential diagnostics of sarcoidosis.